So, I left off talking about epistasis, which is a type of gene that will turn off the expression of other genes. And what I was going to say is that in the case of this hedgehog, it still has genes for hair color and eye color and skin color, but the expression of all of those traits has been turned off because it has the albinism gene to express. So we've talked about epigenetics a little bit already. We watched a video about it in class, but I just want to make sure that you realize that the environment can have an effect on the expression of traits. We see this with sea turtles. Sea turtles inherit the genes to produce both sets of reproductive organs. That means they have both male and female reproductive organ genes, but the ones that get turned on and get expressed are determined by the temperature of the sand when they hatch. Cooler temperatures are female, higher temperatures are male, and because of that, that's how they equalize their population. Then we have Siamese cats. With Siamese coloration, the parts of the body that cool off most or earliest right after birth are the ones that are going to turn this dark color or this offset color compared to the rest of their body, which is this light cream color. So genetics, complex genetics, these are all the types that we've talked about in this video. So you should be aware of these and have these in your notes. But we're going to spend most of this video talking about dihybrid crosses. And what that means is that we're going to be crossing two traits at once. So in a Punnett square, the ones we have done, we call monohybrid crosses because all we're doing is looking at one trait and what the possible offspring would be for that one trait. Now we're going to be looking at two traits at the same time, and that requires a Punnett square box that's 16 boxes big, and we're going to be using two letters, one for each trait. So the first thing to do when we are looking at a dihybrid cross is to figure out the possible gametes that the parents would make. So when we look at a genotype for dihybrid crosses, you will see two letters for each gene. Just because we are looking at two genes at the same time does not mean we will ignore both alleles. So, the best way to think about this is that it's like foiling in math, where you have those two sets of parentheses and you multiply the first of each set. That's what you're going to do to make possible gamete combinations. You're going to start with putting the two first letters together in each half of the genotype. So we're going to take the big A here and the big B here, and those are going to be one possible gamete that this mom could produce. Then we're going to take the outsides. So we take the first letter out front and we take the last letter out back, and those are going to become a possible gamete. So you can see that there. Inside, we're taking the last letter of the first gene and the first letter of the second gene, and now look how they're listed. The first gene is still listed first. The second gene is still listed second. We didn't flip-flop them, even though the first one is a recessive allele. Then we're taking lasts. So we're taking the last allele in each pair. Like I said, it's just like foil. First, outside, inside, last. And then we have the dad's possible gametes. You have firsts, you have outside, you have inside, and you have last. And we're going to take those gametes and we're going to arrange them on our Punnett square. So it's a 4x4 Punnett square, which means there are 16 boxes here. We're still taking one parent and putting it on one side, and the other parent goes on the other side. So you can see how these would be arranged. We have our mom up here, we have our dad along the side, and all we're going to do is fill in the Punnett square. And that's what we see here. Now we have filled in the Punnett square for this dihybrid cross. Okay, so we combined this possible gamete with this possible gamete. Here's the possible offspring that results. And we just do the same thing for the whole dihybrid cross Punnett square. Sorry, my cat's helping me teach you about dihybrid crosses. So, then when we look at genotypes, we have 4 possible genotypes. We have 2 capital A's, so homozygous dominant for the first gene and heterozygous for the second gene. Then we have homozygous dominant for the first gene, homozygous recessive for the second gene. We have heterozygous for both traits, and then heterozygous for the first trait, and homozygous recessive for the second trait. So here are those 4 examples of each of those genotypes and the percentage or the breakdown of their offspring would look, how much of their offspring set would look like that. And then here are the phenotypes. Okay, so 8 out of the 16 would be right-handed and have finger hair. 8 out of the 16 would be right-handed with no finger hair. None of them are going to be left-handed with finger hair, and none of them are going to be left-handed with no finger hair because none of them are going to be left-handed. So this is all we're going to cover with dihybrid crosses. We're going to do a lab where we kind of practice doing this, but make sure that you're kind of understanding how this cross is formed.